So this is practical syllabus, okay, designed by PCI for semester 6th for the subject Herbal Drug Technology. Here, what is the exact purpose to implement such type of experiment in our syllabus? That is question number one. I think you know that very well. The to perform the preliminary, that is initial phytochemical screening of crude drugs means what? What is significance of this type of experiment? What do you mean by
A lot of
phytochemical screening? Yes, we should know the secondary
which
metabolites. Yes, exactly. That is which for the purpose of we know the secondary metabolites, whatever that is present, okay, in our plant material. Okay, when you perform a typical type of experiment, suppose I supply you a powder, okay, and say that you go for the extraction purpose. Okay, you find out whether your powder or your drug contains alkaloids or not. Okay, for that purpose, you go for the phytochemical screening. And phytochemical screening stands for the collection of raw material. Okay, itself it starts from collection of raw material. Actually, it is a very vague process, or you can say that very long process. Collection of raw material up to the what? Up to the biological assay. In that collection of raw material, you have to collect material. Whenever I that is suppose give you a particular proposal, okay, for your UG projects. Okay. To perform phytochemical and pharmacological, that is, for example, a profile of XYZ drug. What is your duty? You have to first find out the whether that plant is located. Okay, that is geographical location. You have to first find out. It is maybe nowadays it is very easy to go to Google and search on that. Okay, from that you go to there that place. You have to collect that material. Okay, then, according to the monograph that will come later on, it is that is monograph analysis means monograph means what? Typical type of that is standardized procedure of that plant material is noted on official books. That is called as a monograph. That is in I think you know that pharmacopoeia means what? Okay, that is example of. Then you should go for the drying, proper drying. Huh? Then after drying, you have to convert that into the powder form. Then after powder form, you go for the what? Yes, different initial screening. Now you decide, you apply, okay, different chemical test for that purpose. Okay, different chemical test for your that powder or alcoholic or aqueous extract. Huh? Suppose all taste that is alkaloidal taste, then tannin taste, glycoside taste. Okay, so, like you know now very well, what are different secondary metabolites? After completion of for applying all these chemical tests, you recognize that your that is powder or your plant material contains, say, suppose flavonoids. Okay, now you targeted go for the flavonoid contents. Okay, then you have to again whatever that is collected plant material subjected for the large scale extraction. Large scale extraction means you have to collect that again powder, then go for the oxidation. And depending on maceration is again, but for the oxidation at the lab level, it is the best method. Okay. In flavonoids, isolation is possible, or that is by oxidation method. So you go for the oxidation. Now here, question is that you directly do not unable to obtain okay flavonoids from that plant material. For that purpose, you go for the successive extraction method. Successive means what? According to the polarity of that plant materials. Okay, means you start from the chloroform and end with the alcohol. So take your plant material. Okay, place in what you can see that muslin cloth or in water paper or you simple a paper. Okay, wrap that powder material and place in symbol of that success material and start oxidation. Okay, after first cycle, suppose you take the chloroform, then whatever that is a residue or you can say that extract that will come in around RBF, then you stop your process. How you can indicate that your process is stopped? When your that is solvent becomes colorless. Okay, that indicate now whatever present in your plant material is now rich, or that will come in your solvent. Okay, then you stop the process, then remove that plant material and dry it for the 24 hours. Okay, then again the same plant material you go for the second solvent. Okay, say for example n-butanol. Again go for the oxidation for complete entire process. Okay, again stop the process, remove that your RVF. Okay, uh, whatever that is extract comes in that, then remove again plant material, again dry it. So like that you continue this process up to the alcohol. Now here what is common? Here common is what you use same plant material, but here what is different? You use different solvent material. So by using this polarity wise, using solvent material and single plant material, that is called as a successive solvent extraction. Okay, and that will help to what exactly isolation of secondary metabolites present in that. It is a very easy process to isolate later on. Okay, so how to it is easy, as we know that in literature, say suppose steroids are there. In literature say that almost all steroids are generally present in pet ether extract. Then you have also pet ether extract. Then you go for the simple pet ether extract and apply the chemical test simple. That is your initial screening. Okay, pet ether extract and go for the steroid test. Okay, Salcosis test is steroid test. Hmm? Lieberman Burchard test is steroid test. And suppose it comes positive, then indicate that whatever you isolated via successive extraction process, it definitely initial stage contain a steroid moiety that you are extract. Then you go for the separation technique. Okay, like that flavonoids again for flavonoid. Here example MCQ may be asked here for your sessional, for your final from this practical point of view. Steroids here I say that what pet ether okay is responsible for the isolation. For flavonoid, for flavonoid ethyl acetate. Okay, you remember this? Huh? Ethyl acetate. Okay, this ethyl acetate solvent is required for isolation of flavonoids. Okay, so majority of flavonoids are come in ethyl acetate solvent. It means that it is not come in alcohol. No, it also come in alcohol, but it becomes very difficult at the time of isolation, separation. That's why we prefer such type of extraction that is called as a successive extraction. Now, question is that what is significance of this incorporation of this practical in your syllabus? So, at the lab level, you should know what type of secondary metabolites okay are there, how these are isolated, okay, what are different chemical tests to be performed for carry out okay these secondary metabolites. Hmm? For that purpose it is implemented. So this is what first your experiment, that is to perform preliminary phytochemical screening of crude drugs. Now here doubt is clear, yes or no?
Yes.
Any problem up to this?
No, sir.
Okay, all of you clear here? Yes.
Sir.
Okay,
Yes.
good.
Yes.
Yes. Next, that is what is second experiment? Second experiment is determination of alcohol content of Asava and Arista. Now, before going to directly Asava and Arista, you should know what do you mean by Asava Arista. This Asava Arista, I think you know that these are nothing but Ayurvedic formulations. Okay, that is intended or used for the purpose to curing particular disease. It contains alcohol. Okay, that is called as a self-generated alcohol. That is called as a alcohol generated, that is nothing but fermentation. And fermentation means what? You know that very well. Okay. So by Asava Arista, there is standard, or we can say that alcohol percentage reported is in a standard, that is in a typical type, typical in percentage. If suppose it exceed, okay, that standard, then its medicinal value becomes very, you know that what is the effect of alcohol. I think we know that very well. Okay, it becomes less. Okay, and other effect starts. That's why you should calculate. You should determine the alcohol content of Asavan Arista. Okay, suppose I complete, okay, a XYZ Asava, whatever that is available in the market, it contains more percentage of alcohol. And instead of giving a therapeutic effect, it gives the sedative effect or other effect. You know that hallucinatory effect. Okay, alcoholic effect is not no. We know that very well. Okay, like that effect. Then what happens? The DI or responsible person collect that sample and analyze in the lab. Okay, for analysis in the lab, you should know what is that process. Okay, for that purpose, at lab level or your degree level, you should know initial, that is idea about the how it is calculated, how it is determined. So this is what purpose of experiment. So determination of alcohol content of Asava and Arista. Now it is clear? Yes or no? Arjuna
Yes, sir.
Arista. Huh? Arjuna Arista is the best that is available. Okay. So like that, Asava Arista, nothing but I think we studied also in second year, or it is not completed. Huh? In second year we studied this. Yes or no?
No, sir.
Okay, no problem. Asava Arista. Wait 2 minutes. Okay. Sorry for the inconvenience cause. So So this is nothing but Asava and Arista. Okay. Next, come to the part that is evaluation of excipient of natural origin. Yes, anyone? Can you tell me some examples of excipient that is obtained from the natural origin? Anyone examples? I think you studied. Yes?
Starch.
Okay, exactly. Starch is very best. That is okay. Definitely, starch is correct answer. Any other? So many natural origins.
Ataka.
Correct, exactly, exactly. And recently, in last month, also one more paper is published. Okay, my paper is published of this particularly use of natural excipients. Okay, in pharmaceutical industry, you can also refer that paper. Okay, and that is published by me along with the PG students. Two students from the Sutics department and one student from the chemistry department. Recently published, that is current, that is publication of mine. Particularly only use of natural excipients. Okay, in pharmaceutical industry. In that, I try to collect all the natural origin excipient, and it's simple. It shows for what purpose it is used. Okay, so you go to the Google and search that. Okay, next, like starch, you know that what is use of starch. Acacia, you know that what is use of acacia. And then cellulose, you know that what is use of cellulose. Okay, pectin, what is use of pectin. So like that. So evaluation of this excipient of natural origin. Evaluation means what? Whether these are fit or not. Okay, for your what as the excipient. Okay, before going to the formulation, you should what evaluate that experiment excipient at the lab level. That is in industry, you have to again evaluate. Okay, this excipient the initial level because it will directly affect on final formulations. Okay, or so it is lost by the formulator, or we can say that that owner. Before that, you should evaluate all that experience. So at the lab level or your degree level, you should know the ideas. Okay, if you enter into the industry, you should know the this basics of this evaluation. Okay, next that is number four, incorporation of prepared and standardized extract in cosmetic formulations like creams, lotions, shampoos, and their evaluation. Here, incorporation of prepared and standardized extract in cosmetic like means what type of hair plants, okay, use in particularly lotions and shampoos. Say, for example, shikakai. Shikakai is a Rita also that is used in what in shampoos very well. We know Brahmi, one of the back of a money. Brahmi Avla Kestel, I think it is very well known. Okay. So like that, you should have prepared and standardized extract. Okay. And then it is incorporated in the different cosmetic formulation, that is lotion, shampoos. So at the lab level, simple, you are able to prepare a lotion, okay, shampoos, syrup, mixture, even the tablets, and their evaluation as per the pharmaceutical requirement. Okay. One more video also available. Okay. I prepared along with our students, okay, your senior students, the preparation of tablets. You go to the YouTube and see there how these tablets, Ayurvedic tablets are prepared. Okay. Already these are those now in final year students. Okay. All these are that is involved in formulation of tablets. Okay. Yes, it is obviously it is available. Go to the Google and add the word that is Firoj Tamboli YouTube only. That is enough for that. Will come definitely. Next, that is number six experiment that is monograph analysis of herbal drugs from a recent pharmacopoeia. Now, monograph analysis of herbal drugs from the recent pharmacopoeia here is simple. And here recently some herbal drugs informations. Okay, it is detail given in the pharmacopoeias. So how it is given, how when it is referred, how it is referred, you should know. Okay, for that purpose, it is that is given or it is incorporated in the syllabus. Okay, so like we know that suppose pharmacognosy of Sena, I ask you directly go to the right from the what synonym up to the allied or you can say that substituent okay of that particular sena but it is officially okay included in monographs how it is studied systematically. So systematic study of any entire or single crude drug that is given in the pharmacopoeia that is called as a monograph analysis of herbal drugs from the recent pharmacopoeia. Okay. Next, number seven, a determination of aldehyde contents. Okay. Now, aldehyde contains, I think you know that in which that is type of secondary metabolites these aldehyde are present. Okay. So for this is nothing but estimation type of okay experiment. You have to calculate aldehyde. Aldehyde it is a functional group. Phenol it is a functional group. Okay. So you have to calculate this percentage, okay, present in your that is fruits, or you can say that in crude drugs, and it is given by systematic procedure. Okay, you have to follow that procedure and simple. Okay, do that experiment. Okay, at the industrial level. So this is again nothing but what we can say that a aldehyde content. Now here, phenol content, again the same phenol content, you have to calculate the phenol percentage of XYZ, that is formulation. And last, that is here determination of total alkaloids. Again, it is one of the what we can say that a very important aspect. Say suppose Vinca is there, Vinca powder is there. You have to go for the calculation of whatever total alkaloids are present. So here is opportunity again. You people like for what you have to again able to what you can say that simple that is publish a paper here possibly, huh? Because to perform the phytochemical screening asavarista you can also do very well here natural origin again do means what aldehyde content phenolic content total here samples preparation whatever you systematically that is mentioned on your that is in your experiment that is nothing but your papers. Okay, that will come into paper form. We have to publish that typical. So think again on that basis. Okay, later on. Next. So I think now up to this, now here clear that why these all this type of experiment is added in herbal drug technology. Now, is any problem up to this?
No, sir.
Okay, exactly. The pattern is like this. Okay. Title of experiment here to perform preliminary phytochemical screening of fruit drugs. Then to determine the alcohol content of asavarista. Number three, to isolate, evaluate the excipient of natural origin. Number four, to prepare and evaluate the herbal poultry. To prepare and evaluate the herbal shampoo. To prepare and evaluate the herbal sunscreen lotion. Sorry. To prepare and evaluate the herbal cough syrup. To prepare and evaluate the herbal mixture. To prepare and evaluate the herbal tablets, to determine the total alkaloids in given crude drugs, to determine the phenol content of crude drugs, to determine the aldehyde content in lemon oil, and to study the monograph analysis of herbal drugs from Indian pharmacopoeia and herbal drugs from the Siddha pharmacopoeia. Here, 14 experiments are here I designed, okay, particularly. See, how much that is we are able to cover this one. So come to the first that is experiment number one to perform the preliminary phytochemical screening of crude drugs. Here requirement test tube stand, test tube holder, water bath, filter paper, pipette, gas, burner, extract of crude drugs. Now here you know that what is exact purpose of this experiment. Now it's clear, I think. Okay, whatever I explained initial, that is nothing but this introductory part. Here chemicals, all these chemicals are required. That is hydrochloric acids, then dragon drop reagents, Mayer's reagents, Wagner's reagents, Hager's reagent, H2SO4, okay, 1% gelatin solution, sodium chloride, bromine solution, concentrated HCl, magnesium turnings, alpha naphtol solution, Benedict reagents, swelling reagents, A and B, concentrated nitric acid, nitrogen reagents. Okay. In introduction here, that is whatever I explained, that is you quoted again in your language in intro part. Then come to the qualitative